Welcome back friends, welcome again another video tutorial of A to Z biology. Today's topic is genetical basis of cancers and how cancer is developed, with the basic characteristics of the cancer cells and the proto-oncogenes and tumor suppressor genes. So basically today's topic is about proto-oncogene and tumor suppressor gene. We all know that what is cancer. What is cancer? We all know that what is cancer. Cancer is abnormal cell growth, and cells grow abnormally, cells grow rapidly, cells proliferate, and cell ultimately form a mass of tissue called tumors. Benign tumors are not called cancers. Tumors which can invade the surrounding tissue and cells will be able to metastasize. Metastasis means this is the tumor forming cells. And cells will be able to move from one side into another side. One side to another side. These properties called metastasis, and these properties only shown by malignant tumors. These types of tumors are actually cancers. This is the basic definition of cancers, how cancer is formed. Cancers, due to different kinds of cause, cancers will be developed due to radiation, due to chemical carcinogenesis, due to chemical carcinogens. Radiations and chemical carcinogens leads to cancers. They actually causes cancers, and the viral infections causes cancers. Different kinds of infections actually activate the oncogene. Today's main discussion is about oncogene and proto-oncogene. Proto-oncogene and oncogenes. Onco word is related to cancer. Oncology means the study of cancer. Proto-oncogenes and oncogenes. What is proto-oncogenes? The basic and very basic definition of the proto-oncogene is that proto-oncogene is a gene. Proto-oncogenes are group of genes which produces different kinds of growth factors. These proto-oncogenes are not related to cancers. Proto-oncogenes are normally present in our body, present in our each and every cells. Proto-oncogenes are present. These proto-oncogenes produces different kinds of growth factors. GF stands for growth factors, different kinds of transcription factors, different kinds of signaling molecule which actually involved in different kinds of signaling, and GPCR, G protein coupled receptors, and G proteins, and GTPases, and other kinds of proteins which actually involved in cellular differentiation, cell growth. These are called proto-oncogenes. This is a counterpart of oncogenes. Normally proto-oncogenes is present within us, but suppose this gene product is mutated, so proto-oncogene normally produces these different kinds of proteins, growth factors, transcription factors, signaling molecule, GPCR, G proteins, normally produces these kinds of proteins. They help in their function is that they actually increases cell proliferation, cell division, Now you think about that, when this proto-oncogene is mutated, when this proto-oncogene is activated, when this proto-oncogene is converted into oncogenes, this proto-oncogenic product will be increased. These oncogenes will be produce more and more amounts of growth factors, more and more amounts of transcription factors, more and more amount of signaling molecules, GPCR, other kinds of proteins that actually involve in cellular differentiation and cellular growth. So that condition actually increases the cell growth. So increasing cell growth actually leads to cancer. Abnormal proliferation of the cell develops tumor, at first develops tumor, then cancers. So proto-oncogene is sometimes converted into oncogenes. In case of cancer patients, we see that proto-oncogene is converted into oncogene in different kinds of cancers. In different kinds of cancers, proto-oncogene is converted into oncogene due to mutation or activation. This proto-oncogene is a normal gene present within us, and this proto-oncogene is converted into oncogene, and this oncogene produces oncoproteins. These are natural growth factors, transcription factors, but the production of oncoproteins is very high. So the cell grow very high, cell differentiate very high, cell proliferation will be high. That leads to cancer. But oncogene not only originated from the proto-oncogene, oncogene comes from other sources like different kinds of viruses. Viruses carry oncogenes also. Viruses carry oncogenes also. The first virus which is detected or which is discovered carrying oncogenes is RSV virus, Rous sarcoma virus. This is the virus which carries oncogene. Other kinds of viruses also carries oncogenes, Epstein-Barr virus, EBV, Epstein-Barr virus, and these kinds of viruses carries oncogenes. These viral oncogenes, these viral oncogenes is transmitted into host cells. Here actually host cells is converted into cancerous cells due to transmission of these viral oncogenes, or any kind of viral genes or viral genes or viral enhancer genes or promoter gene also leads to activation of some oncogenes in the host cell. How it is possible? Promoter region of a viral gene is entered into our host cell, and they get stick with the gene which produces lots of proteins. They get stick. Suppose this is a viral gene. This viral gene is stick with the normal host cell gene. This is normal host cell gene. Due to viral infection, this gene is stick with the host cell gene. This gene normally produces lots of protein, and you think that this viral enhancer increases the expression of this gene. That also leads to production of more and more amount of growth factors. That also leads to excessive cellular divisions. Now this is the proto-oncogene, oncogene. Different kinds of proto-oncogenes are discovered. Most important proto-oncogene is RAS. RAS have different subform: KRAS, HRAS, and RRAS. This is the different kinds of RAS. MYC. This is the two important type of proto-oncogene. If this gene is mutated, gene mutation is due to point mutation. This is the different kinds of mutation. Point mutation, chromosomal translocation, point mutation, chromosomal translocation and deletion, duplication. This is the different kinds of mutations. Gene amplification, another is gene amplification. This is the different kinds of events actually converts a proto-oncogene into oncogenes, and oncogenes produces lots of oncoproteins. Chromosomal translocation is seen in Burkitt's lymphoma, jaw cancer. Chromosomal translocation. In this translocation, in this event, a piece of chromosome is translocated from one chromosome into another chromosome. This is the different kinds of proto-oncogene. When this proto-oncogene is mutated, this proto-oncogene form oncogene. Oncogene produces lots of oncoproteins which eventually leads to cancer. RAS produces a GTPase. You know about GTPase? What is GTPase? This RAS protein encodes a protein which is called GTPase. This GTPase protein involved in G protein signaling, GPCR signaling. G protein signaling, you know G protein signaling. This is a cell surface receptor. This is GPCR, and different kinds of protein is attached with this receptor is called G protein. G protein has different subunit alpha, beta, gamma. Alpha have binded with GDP. When a extracellular signaling molecule comes, this GDP subunit is exchanged for GTP, and this G protein is activated. This G protein activates a membrane spanning enzyme called adenyl cyclase. And after this activation, this G protein is inactivated by this GTPase. GTPase break down into GDP, and this G protein is inactivated, so signaling will be halted. But in case of RAS mutation, this GTPase will not be coded by the RAS gene, or GTPase is defected, so GTPase activity is not present. So G protein signaling will be activated but not inhibited. So different kinds of cell signaling pathway like MAP kinase pathway and other cell signaling pathway will be activated, which leads to excessive cell proliferation and excessive cell division. Another is MYC gene. MYC gene codes for transcription factor, which regulates the cell cycle. Transcription factor, activating transcription factor, which regulates the cell cycle, and different kinds of regulatory proteins present in the cell cycle. If this gene is mutated, if this proto-oncogene is mutated and form oncogene, meek oncogene produces lots of transcription factors which increases the cell progression through different kinds of stages, G1, S, G2M. So these are the proto-oncogenes. These are the proto-oncogenes. Another kind of genes which play important role in cancer formation is tumor suppressor gene. Other kinds of proto-oncogenes are present. Almost 100 types of proto-oncogenes are present, but this is the two main type which is important. Another kind of genes which actually plays role in cancer is tumor suppressor gene. Tumor suppressor gene plays a key role in cancer. How? The name indicates that normally this gene actually inhibits the formation of any kind of tumors. This gene actually inhibits the excessive cell proliferation, excessive cell growth. These are called tumor suppressor genes. This is the most beneficial genes which actually inhibits the formation of cancer. But if this gene is mutated, if this gene is inactivated, then cancer will be formed. But remember that proto-oncogene mutation in the one allele of the proto-oncogene. Genes have two alleles, pairs of genes. two alleles. Alleles are pairs of genes present on the homologous chromosome on the same locus which produces a protein product which determines a specific trait. They are called alleles. So proto-oncogenes, mutation in one allele of a proto-oncogene leads to oncogenic effects, but mutation in both of the alleles of the tumor suppressor gene can cause cancer. Not one allele, both of the allele. But this type of mutation is inherited. Proto-oncogene mutations is occurred in somatic cells, so it is not inherited. But this gene mutation may be occurred in germinal cell or it may be occurred in somatic cells. So this kind of mutation may be inherited. This is the basic difference. And tumor suppressor gene, the most important tumor suppressor gene is P53. Sorry, P53, TP53. TP53, it is the gene, most important tumor suppressor gene. Another is BRCA, breast cancer gene. Another is RB gene. Other genes are WT, Wilms tumor related factor, WT, APC, other kinds of genes are also present, P10, other kinds of genes. Another are SMAT4. These are the different kinds of genes, tumor suppressor genes. The main tumor suppressor gene is TP53 and RB. TP53 gene and RB. RB is the first identified or first discovered tumor suppressor gene, retinoblastoma gene. If this gene is mutated, if this gene is altered, if this gene is undergoes different kinds of mutations, they leads to retinoblastoma. They leads to retinoblastoma. BRCA, breast cancer genes, if this gene is mutated, they develop breast cancer. Most important is tumor TP53. Tumor suppressor genes have different kinds of function. The most important function is gatekeeper function and caretaker function. Gatekeeper function actually allows apoptosis, allows the arrest of cell cycle and cell proliferation, also controlled by the gatekeeper function of this tumor suppressor gene. But caretaker function of the tumor suppressor gene, it actually allows actually checks the DNA, if DNA is damaged or not. If the cells, please remember cell cycle, G1 to S, G2 and to M, this is the cell cycle, this is interphase, this is mitosis. If the DNA is damaged inside the cell and cell is entered from G1 into S and the DNA is duplicated, and that leads to mutation in further generations. So caretaker function of the gene is also important. So these two types of functions are played by the tumor suppressor genes. RB gene produces a protein, RB gene produces RB protein which restrains the passage of cell from G1 to S phase. The RB protein is a transcription repressor, binds with E2F protein which is called transcription activator, and inhibits the E2F. Therefore, cell will not be allowed to G1 into S phase. So inhibition of this gene or mutation of this gene leads to retinoblastoma. Another most important tumor suppressor gene is P53 gene. P53 is called genome guardian or molecular policeman. P53. P53 is not actually is a protein. It is coded by TP53. TP53. When DNA is damaged, any kind of stress, any kind of harmful radiation, any kind of agents which actually damage the DNA, they actually activates the P53. P53 activates DNA repairing genes, or some cases DNA is very damaged, the different portions of the DNA is damaged and cell will not properly correct the DNA damage, so cell will commit suicide via apoptosis. So apoptotic pathway is also induced by these genes. So this gene is very much regulated and this gene is highly important to us. This gene is present in the chromosome number 17 in the P arm. So TP53 gene is present on the chromosome number 17 in the P arm. P arm is the short arm of the chromosome. So TP53 codes for P53 protein. This is a gene codes for P53 protein. Normally all the times this protein is present inside the cell, and this protein is deregulated by MDM2 protein. MDM2 binds with the P53, binds with the P53 and degrades the P53, and P53 MDM2 complex will be formed. MDM2 complex will be formed and P53 is degraded. Cellular P53 level will be low. Cellular P53 level is very low due to degradation by MDM2. Normally this is occurred, but if there is damage in the DNA, this is DNA, and any kind of stimulus, any kind of agent which damages the DNA damage, any kind of stimulus which actually damages the DNA activates the P53. How it is possible? Any kind of stimulus which actually damages the DNA damage leads to phosphorylation of the P53 protein and MDM2 on their binding domains, so they no longer be able to bind with each other. So P53 degradation is not occurred due to phosphorylation. So P53 level inside the cell is decreased or increased? Increased. P53 protein level is increased inside the cell, inside the cytosol. Now this P53 acts as a transcription factor. This P53 increases gene transcription, a specific kind of gene which is called WF1. WF1. This is a specific kind of gene, and P53 acts as a transcription factor. This P53 increases the transcription of WF1, and WF1 codes for a protein called P21 protein. And this P53 this P21 protein, this is a protein coded by WA1 genes, and this P53 also acts as a transcription factor and increases the transcription of DNA repairing genes. DNA repairing genes, because DNA is damaged, we need to fix that damage by increasing the synthesis of DNA repairing proteins. So DNA repairing gene transcription will be also increased. WF1 gene transcription leads to P21 protein production. This P21 protein binds with CDK4 cycline cyclin D complexes. This P21 protein binds with CDK4 and cyclin D complexes and inhibits the kinase activity of these complexes. Normally these complexes actually phosphorylate other kinds of proteins. So P21 binding inhibits the phosphorylation of these complexes and inhibits the cell cycle progression from G1 into S phase. We know the cell cycle. Cell first enters into G1, then enters into S phase, then enters into G2 phase, then enters into M phase. So this P21 when binds with the CDK4 cyclin D complexes, they actually inhibits the cell at G1. Cell enters the G1, does not enters the S phase, because different kinds of other proteins which actually helps in this phase transition is not phosphorylated, like PRB2, E2F complex will not be formed due to unphosphorylation of this molecule. So cell will be arrested at G1 phase. But if you think this gene is mutated, please think about that. This gene is mutated and this protein is not formed. Cell which is entered into G1 entered into S phase with damaged DNA. If DNA is damaged, this P21 protein is not produced, because this P53 gene is mutated, this P53 gene is inactive. So cell enters G1, enters S phase, G2 phase, M phase, and cell cycle is progressed, cells divides, lots of cells will be produced and the cells becomes cancerous. Another thing about the P53 gene is that they actually, if the DNA damage is not repairable, P53 gene start apoptotic pathway. Apoptosis is programmed cell death. Cell commits suicide. Suicidal pathway or death pathway is promoted by the P53. They activate the BAX gene, BAX, and this BAX gene codes for BAX protein. This BAX protein inhibits a repressor called BCL2. This BCL2 is a repressor of this apoptotic pathway. This is a repressor protein of this apoptotic pathway. This BAX protein inhibits the BCL2 expression. Therefore, apoptotic pathway will be increased. Apoptosis will be increased. P53 normally inhibits the expression of oncogenes, normal cellular oncogenes, normal proto-oncogenes to normal cellular proto-oncogenes to oncogenes, and other kinds of viral oncogenes into oncogenes. Thank you for watching this video. If you like this video, please subscribe my channel.